In this lesson, we will learn about nature of enzyme. Introduction. The living cell is the site of tremendous biochemical activity called metabolism. This is the process of chemical and physical change which goes on continually in the living organism. Build-up of new tissue, replacement of old tissue, conversion of food to energy, disposal of waste materials, reproduction, all the activities that we characterized as life. Building up and tearing down takes place in the face of an apparent paradox. The greatest majority of these biochemical reactions do not take place spontaneously. The phenomenon of catalysis makes possible biochemical reactions necessary for all life processes. Catalysis is defined as the acceleration of a chemical reaction by some substance which itself undergoes no permanent chemical change. The catalysts of biochemical reactions are enzymes and are responsible for bringing about almost all of the chemical reactions in the living organisms. Without these enzymes, the reactions take place at the rate far too slow for the pace of metabolism. The oxidation of fatty acid to carbon dioxide and water is not a gentle process in a test tube. Extremes of pH, high temperatures and corrosive chemicals are required. Yet in the body, such a reaction takes place smoothly and rapidly within a narrow range of pH and temperature. In the laboratory, the average protein must be boiled for about 24 hours in a 20% HCl solution to achieve a complete breakdown. In the body, the breakdown takes place in 4 hours or less under conditions of mild physiological temperature and pH. It is through attempts at understanding more about enzyme catalysts, what they are, what they do and how they do it, that many advances in medicine and the life sciences have been brought about. We will have a glance over some early enzyme discoveries. The existence of enzymes has been known well for over a century. Some of the earliest studies were performed in 1835 by the Swedish chemist John Jacob Berzelius, who termed their chemical action as catalytic. The first enzyme was obtained in pure form in 1926 by James B. Sumner of Cornell University. Sumner was able to isolate and crystallize the enzyme urease from the jack bean. For his work, he earned the 1947 Nobel Prize. In sharing with John H. Northrop and Wendell M. Stanley of the Rockefeller Institute of Medical Research, Northrop and Stanley discovered a complex procedure for isolating pepsin. This precipitation technique has been used to crystallize several enzymes. Let's explore the chemical nature of enzymes. All known enzymes are proteins. They are high molecular weight compounds made up of chains of amino acids linked together by peptide bonds. Enzymes are proteins made up of amino acids. Active sites are formed by secondary and tertiary clustering of amino acids, which results in the formation of physical grooves where the substrate binds. Charges on amino acids allow them to interact with substrate by donating or accepting electrons or protons depending on the enzyme. Enzyme substrate binding involves temporary H positive and or ionic bond at the active site. Enzymes can be denatured and precipitated with salts, solvents and other reagents. They have high molecular weights ranging from 10,000 to 20 lakhs. Many enzymes require the presence of other compounds, that is cofactors. Before their catalytic activity can be exerted, this entire active complex is referred to as holoenzyme. That is, apoenzyme means the protein portion and the cofactor means the coenzyme, prosthetic group or metal ion activator. According to Holm, the cofactor may be, first one is coenzyme. It is a non-proteinaceous organic substance which is thermostable and loosely attached to the protein part. Second is prosthetic group. It is an organic substance which is thermostable and firmly attached to the protein or apoenzyme portion. Third one is metal ion activators. It includes ions like K positive, Fe2 positive, Fe3 positive, Cu2 positive, CO2 positive, Zn2 positive, Mn2 positive, Mg2 positive, Ca2 positive and MO3 positive. Now we will study naming and classification of enzymes. Enzyme Commission has developed a rule for naming enzymes. According to this rule, each enzyme is classified and named according to the type of chemical reaction it catalyzes. The Enzyme Commission has given each enzyme a number with 4 parts like EC 2.7.1.2, that is hexokinase. The first 3 numbers define major class, subclass and sub-subclass respectively. The last number in a serial number is the sub-subclass, indicating the order in which each enzyme is added to the list. Common name and EC numbers of some enzymes are alcohol dehydrogenase, EC number 1.1.1.1, phosphofructokinase, EC number 2.7.1.11, glutamine synthetase, EC number 6.3.1.2, acetylcholinesterase, EC number 3.1.1.7. Except for some of the originally studied enzymes such as pepsin, renin and trypsin, most enzymes names end in ASE. The International Union of Biochemistry initiated standards of enzyme nomenclature which recommended that the enzyme names indicate both the substrate acted upon and the type of reaction catalyzed. Under this system, the enzyme uricase is called urate O2 oxidoreductase, while the enzyme glutamic oxaloacetic transaminase is called L-aspartate 2-oxaloglutarate aminotransferase. Enzymes can be classified by the kind of chemical reaction they catalyzed. First one is addition or removal of water, hydrolysis. These include esterases, carbohydrases, nucleases, deaminases and proteases. Hydrases such as fumarase, enolase, acotinase and carbonic anhydrase. Second is transfer of electrons from oxidases, dehydrogenase. Third one is transfer of radical, transglycosidases of monosaccharides, transphosphorylases and phosphomutases of phosphate group, transaminases of amino group, transmethylases of methyl group, transacetylases of acetyl group. Fourth is splitting or forming a carbon-carbon bond, desmolases. Fifth one is changing geometry or structure of molecule, isomerases. Last one is joining two molecules through hydrolysis of pyrophosphate bond in ATP or other tri-phosphate, ligases. Moving ahead to enzyme kinetics. Here we will firstly study basic enzyme reactions. Enzymes are catalysts and increase the speed of chemical reaction without themselves undergoing any permanent chemical change. They are neither used up in the reaction nor do they appear as reaction products. The basic enzymatic reaction can be represented as substrate plus enzyme gives rise to product plus enzyme. A theory to explain the catalytic action of enzymes were proposed by the Swedish chemist Svante Arrhenius in 1888. Proposed that the substrate and enzyme formed some intermediate substance which is known as the enzyme substrate complex. The reaction can be represented as substrate plus enzyme gives rise to enzyme substrate complex. If this reaction is combined with the original equation 1, then the reaction becomes substrate plus enzyme gives rise to enzyme substrate complex, which again gives rise to product plus enzyme. Two important models have been proposed to describe the binding process of enzyme to substrate. The first, lock and key model, proposed by Emil Fischer in 1890. According to this model, the active site of unbound enzyme is complementary in shape to that of substrate. That is, the specificity of binding depends on the precisely defined arrangement of atoms in an active site. To fit into the site, a substrate must have a matching shape. However, it is now evident that the shape of the active sites of many enzymes are markedly modified by the binding of substrate, as was postulated by Daniel E. Koshland Jr. in 1958. The active sites of these enzymes assume shapes that are complementary to that of the substrate only after the substrate is bound. The process of dynamic recognition is called induced fit model. Let's see how it works. Enzyme substrate interaction involves multiple bonds, individually weak, readily reversible. Upon substrate binding, enzyme changes conformation at active site to promote catalytic function. Change in conformation ensures specificity to fit, also allows for the activation of substrate. Three mechanisms by which this occurs are bond distortion, enzyme donates or accepts protons, enzyme donates or accepts electrons. Temporary bonds between substrate and enzyme form transient enzyme substrate complex. Intermediate step in catalytic event, product then released, enzyme reverts to initial conformation. Now let's discuss specificity of enzymes. One of the properties of enzymes that makes them so important as diagnostic and research tools is the specificity that exhibit relative to the reaction they catalyze. A few enzymes exhibit absolute specificity, that is, they will catalyze only one particular reaction. Other enzymes will be specific for a particular type of chemical bond or functional group. In general, there are four distinct types of specificity. The first one is absolute specificity. The enzyme will catalyze only one reaction. Second is group specificity. The enzyme will act only on molecules that have specific functional groups such as amino, phosphate and methyl groups. Third is linkage specificity. The enzyme will act on a particular type of chemical bond regardless of the rest of the molecular structure. And the last is stereochemical specificity. The enzyme will act on a particular steric or optical isomer. Though enzymes exhibit great degrees of specificity, cofactors may serve many apoenzymes. For example, nicotinamide adenine dinucleotide is a coenzyme for a great number of dehydrogenase reactions in which it acts as a hydrogen acceptor. Among them are the alcohol dehydrogenase, malate dehydrogenase and lactic dehydrogenase reactions. Knowing about energy levels. Chemists have known for almost a century that for most chemical reactions to proceed, some form of energy is needed. They have termed this quantity of energy the energy of activation. It is the magnitude of activation energy which determines how fast the reaction will proceed. It is believed that enzymes lower the activation energy for the reaction they are catalyzing. The enzyme is thought to reduce the path of the reaction. This shortened path would require less energy for each molecule of substrate converted to product. Given a total amount of available energy, more molecules of substrate would be converted when the enzyme is present, that is the shortened path, than when it is absent. Hence, the reaction is said to go faster in a given period of time. Elaborating factors that affect enzyme activity. Several factors affect the rate at which enzymatic reaction proceed, like temperature, pH, enzyme concentration, substrate concentration and the presence of any inhibitors or activators. First one is effect of temperature. Since most of the enzymes are proteins, so they unfold and denature at high temperature. This denaturation causes a decrease or loss of catalytic activity, which in turn decreases the rate of reaction. Similarly, low temperature also inactivates the enzyme. Within limits, the rate of an enzyme catalyzed reaction increases with increase in temperature because the greater kinetic energy of both enzyme and substrate molecule ensures more frequent collisions. Thus, an enzyme catalyzed reaction achieves its maximum reaction at an intermediate temperature called optimum temperature. The optimum temperature varies greatly from enzyme to enzyme and especially from organism to organism. The reaction rate of most enzymes of homotherms are maximum at about 40 degrees centigrade. Some enzymes retain activity at unusual high temperature. For example, enzymes like Taq DNA polymerase present in thermophilic bacteria Thermus aquaticus has optimum temperature 72 degrees centigrade. Second is effect of pH. Enzymes are sensitive to pH. This pH dependence is due to the presence of charged amino acids at the active site. Variation in pH cause changes in overall charge of amino acids. Thus, enzymes have its optimum pH at which rapid activity occurs. For example, optimal pH for pepsin, that is protein digesting enzyme, is 2 and for trypsin it is 8.5. Optimum activity is generally for most of the enzymes are observed between pH values 5 to 9. When the activity is plotted against pH, a bell-shaped curve is usually obtained. Third is enzyme concentration. In order to study the effect of increasing the enzyme concentration upon the reaction rate, the substrate must be present in an excess amount, that is, the reaction must be independent of the substrate concentration. Any change in the amount of product formed over a specific period of time will be dependent upon the level of enzyme present. Fourth is substrate concentration. If we examine the effect of varying the substrate concentration on the rate of enzyme catalyzed reaction, when enzyme concentration is kept constant, it is found that at relatively low concentration of substrate, the rate of reaction increases in proportion to the concentration, but as the concentration is increased further, the reaction rate is increased by smaller and smaller amounts. Finally, when the substrate concentration is raised still higher, a point will be reached beyond which there is no further increase in the reaction rate. At this stage, even if the high substrate concentration is added, the reaction rate will remain essentially constant. At this stage of reaction, it is called the maximum velocity or maximum rate, which is designated as Vmax. All enzymes exhibit the saturation effects. A general theory of enzymes was proposed by Michaelis and Mentene, who also proposed the mechanism of formation of enzyme substrate complex on this basis. From the saturation curve, an equation was derived which is known as Michaelis constant, which specifies the quantitative relationship between substrate concentration and Vmax for different enzymes. It is defined as the substrate concentration at which a given enzyme yields 1/2 its maximum velocity, V by 2 equals to Km. Value of Km is important in tending out the affinity of enzymes with that of substrate. Lower the Km values, higher the affinity. Higher the Km values, lower the affinity. This relates relationship between substrate and enzymes. The Km values of different enzymes differs depending upon the condition at which enzymes reaction takes place. It is one of the important characteristic of enzymes. The characteristic shape of the substrate saturation curve for enzymes can be expressed mathematically by Michaelis-Mentene equation. Here, Vmax represents the maximum rate achieved by the system at maximum substrate concentration. The Michaelis constant Km is substrate concentration at which the reaction rate is half of Vmax. Biochemical reactions involving a single substrate are often assumed to follow Michaelis-Mentene kinetics without regard to the model's underlying assumptions. Equation describing relationship between rate of reaction and substrate concentration for enzyme catalyzed reaction. And lastly, we discuss enzyme inhibition. Inhibition of enzyme activity may be irreversible or reversible. First one is irreversible inhibitors. Irreversible inhibitors usually bound covalently to the enzyme and destroy the functional group and active site. Most of irreversible inhibitors are toxic substances. The action of nerve gases on acetylcholinesterase, an example that plays an important role in the transmission of nerve impulses, is an example of irreversible inhibition. Second is reversible inhibitors. In reversible inhibition, the inhibitor can dissociate from the enzyme. Reversible inhibitors involve the non-covalent binding with enzymes. Two common types of reversible inhibitors are: the first one is competitive inhibition. The structure of competitive inhibitors closely resembles that of the enzyme's normal structure. Example: in case of methanol poisoning, methanol in the body is converted to harmful formaldehyde by alcohol dehydrogenase. A high dose of ethanol is used to alleviate the effect of methanol because ethanol competitively binds with the active site of alcohol dehydrogenase. Second is non-competitive inhibition. In non-competitive inhibition, the inhibitors binds to the enzyme at a site other than the active site. Inhibitors binding alter the enzyme's three-dimensional configuration and blocks the reaction. Metal ions including copper, mercury and silver act as a non-competitive inhibitors of many enzymes by binding reversibly to the SH group of cysteine, the amino acid that forms disulfide bridges. This disrupts the structure of enzyme.
Thus, at the end, to summarize, the chapter started with introduction to early enzyme discoveries, followed by understanding of chemical nature of enzymes. Naming and classification of enzymes according to the rules of enzyme commission and the type of chemical reaction they catalyzes has also been studied and cited. Enzyme kinetics with its reaction has been elaborated. Specificity of enzymes, a very important diagnostic and research tool, is also discussed. Energy levels have been described specifically as they are very important for most of the reaction to proceed. Several factors that affect enzyme activity like temperature, pH, enzyme concentration, substrate concentration and the presence of any inhibitors or activators have also been mentioned. Lastly, we have seen enzyme inhibition with its types and examples.